Welcome to the Jack Westin MCAT Podcast with your host, Phil Hawkins.
And Azaii Muñiz.
Welcome back to yet another episode of the Jack Westin MCAT Podcast. So we get to continue the very exciting task of working through the science section, namely that we're working through the bio and biochem section of the MCAT exam. So today we are going to talk a little bit about two very important organ systems. And I personally like them. And if that doesn't show, I've done my job wrong. So we're going to be talking about the intersection between the nervous system and the musculoskeletal system. So today we're going to be talking about how your muscles work. So a little bit of background, right? Yes, you know that you need to know that your muscles, right, they contract, but you also need to know how they get that signal. How do they know to contract, right? And so our goal today is just kind of touch base on that a little bit, and then talk through what it actually means when they contract. And of course, some of the ramifications, some of the consequences when things go wrong. So with the thinking about how the muscles get the signal to contract. So we know that a lot of what goes on in our body is signaled through neurotransmitters. Right? The nervous system, the nerves, the neurons in our bodies will signal their intent with chemicals called neurotransmitters. One of the most important ones in your body is acetylcholine. And so it plays this really big part in muscle contraction and muscle signaling. So when the presynaptic neuron, so you have this neuron, right, that is synapsing with a muscle, it means it's the neuron that is going to signal to the muscle. When we talk about the neuron that is doing the signaling, we call that the presynaptic neuron. So when that neuron gets a signal, right, it has the action potential run down its axon, run down that neuron, and there's going to be a voltage-gated calcium channel. So this very big important distinction, you can have something that is voltage-gated or ligand-gated. So you can pause, try and think about what that is, and then come back. Okay, so with voltage gated, it means that there is a change in there's a difference across a membrane, right? In the voltage, right? The electrical current across the membrane in this case. If it's ligand gated, it means I can give it a physical ligand, right? Physical signaling molecule, and it will respond. So my ligand is going to be my non-existent cup of tea, and I give it to Phil, and then Phil's like, oh, okay. Well, I'm going to do something with it.
We'll do something, right. I always like to think of the ligand as like a key, right? Mm-hmm. Like it's a key to unlock this gate or this door. So now stuff can kind of like move through kind of overall.
My morning key is definitely tea.
Yeah, there you go. Yeah.
So now you have these voltage-gated calcium channels in the neuron. And so they sense that, hey, we received a signal. We need to release the neurotransmitter acetylcholine. So they let calcium in. Now calcium is inside this neuron, and it's going to interact with some proteins that are going to allow that vesicle, right? That little, that ball, so to speak, of holding the neurotransmitters, right? To fuse with the membrane at the end of the neuron and release the neurotransmitters. So far, we've gotten signal to the neuron. The neuron has a voltage difference across its membrane. Calcium comes in, binds within the neuron, and allows the neuron to release acetylcholine, which is the neurotransmitter that the muscle is going to respond to. So the muscle sees this, and they have acetylcholine receptors at their part of the synapse, right, where the two are meeting. And so that acetylcholine is going to bind. So now if we've had acetylcholine bind to the receptor and the receptor is responding, I want you guys to think about, okay, what type of channel is that, right? So that's going to be ligand gated because it needs the ligand, it needs that specific molecule in order to respond. So now you have the acetylcholine, and it's going to cause the opening of sodium channels. Some of this should be ringing a bell because you've seen a lot of similarities with just nervous system signaling in general, right? So now the sodium channels open, and so there's going to be this action potential across the sarcolemma, the muscle cell. This is going to cause yet another release of signals. So in this case, calcium. So now there's calcium inside the muscle, and this calcium is going to be important for the muscle contraction. It's one of the key components that allows for muscle contraction. Now, there is so much more that we could go in depth here, but at some point you need an image, right? You're probably trying to picture some of this and you're like, okay, Azaii, like you're starting to lose me. So I don't want to lose you guys. I want you to go and grab a visual of what muscle contraction itself actually looks like once it gets the signal and once it has that calcium flood in, right?
Right. Yeah, it gets way more complex inside the muscle because there's like troponin and tropomyosin and calmodulin and like all of those things. But like right now, just because there's some very interesting diseases and disorders and things that are related to this neuromuscular junction. It's just really important to understand like the signal in the neuron opens coming down, opens a voltage gated calcium channel. Calcium runs in, makes it release acetylcholine. The acetylcholine binds the receptor on the neuron or the muscle, which opens a channel for sodium. And so it's a ligand-gated sodium channel. Sodium rushes in, that depolarizes the muscle cell. Turns out muscles can depolarize just like neurons. And when that muscle cell depolarizes, it opens calcium channels. So in both cases, the depolarization of the neuron and the depolarization of the muscle are both opening calcium channels. And so when the signal comes down through the axon, the calcium channel opens in the neuron, releases acetylcholine that depolarizes the muscle, which opens the calcium channel in the muscle cell. And so it's like a super interesting thing that like calcium is playing a big role in this. And the MCAT will totally expect you to understand this, like what's going on with like the calcium channels and the neuron and in the muscle, as well as the ligand gated channel. And that like that vocab of voltage gated versus ligand gated is something that is like the MCAT tests a ton as well. And so kind of like what opens these channels, is it like just some change in the charge? Most of the time, just as a step back for a second, most of the time cells are going to be more negatively charged inside and more positively charged outside. And so we tend to have sodium on the outside and calcium on the outside. And we kind of like pump those things to those sides. And so like when we open a channel, those things tend to rush in and that makes it positively charged. And so that's why they're depolarizing. But yeah, this whole, like everything that we're about to talk about with these really interesting toxins and disorders are kind of built around understanding how this neuromuscular junction works. I do think there is one other thing that's kind of interesting here. So acetylcholine is what kind of triggers this, like release from the neuron. And so like a good question is, how do we stop a contraction, right? Like I can like flex a muscle and then I can stop flexing it, right? And so how do we stop that contraction is we just destroy the acetylcholine. So there's an enzyme called acetylcholinesterase. If you ever see something that ends in ACE, that means it's an enzyme. Like lactase is the enzyme that cuts up lactose, and maltase cuts up maltose, and nucleases cut up nucleotides. Proteases are proteins that cut up other proteins. And so acetylcholinesterase is an enzyme that chews up acetylcholine. And so you destroy that acetylcholine and that stops the signal, and that's how you stop the contraction. I feel like it's super interesting, like to realize that all of that is happening anytime you flex a muscle and relax it. And you like think about somebody like playing the piano, like how many times they have to like fire different muscles and how quickly they need to be able to like flex this muscle and then relax it. And like that like hyper controllability is really just controlled by like releasing a little bit of acetylcholine and then chewing it up, and like how this is controlled. And it's super interesting just kind of overall seeing like that work in like a real world. Like this is how I walk. This is how I play the piano. This is how I scratch my head, is like I'm able to like move my body around by flexing different muscles. And we have a whole lot of control. And so I think it's just worth it to just take a second and just appreciate how freaking cool it is that we can like do like really fine, very quick muscle movements and have a whole lot of control over that.
And the crazy thing is, even something as simple as like moving your eyes, right? I don't know if you've ever seen someone try and just follow someone else's finger with just their eyes. You can see, depending on the person, what conditions they may or may not have, you can almost see like the tiny little movements, right? Mm-hmm. And you think about that. And so there's this voluntary control. And then there's also these involuntary movements that sometimes people have that may be normal, that may not be normal. And it's happening so fast. And it's such a really cool thing to think about and have the opportunity to be like, I know why that happens.
Yeah. Yeah. It is worth mentioning that they're like, what we're talking about right now is like skeletal muscle. Compared to like smooth muscle or cardiac muscle, they're controlled by slightly different processes. They have what they call myogenic responses are like myo, meaning muscle, genic, meaning generated. So they have muscle generated activity. And so your heart muscles will contract kind of on their own, and like smooth muscle will as well without getting that signal. But skeletal muscle, like how we like walk and move and flex our biceps, and suck in our stomach when somebody pretty walks by, that is all controlled by like this neuromuscular junction, like the signals coming from the brain going to those skeletal muscles.
And just a quick reminder, there are a lot of nerves that innervate different muscles, including like, the heart and your GI system. But for what we're talking about here, like Phil pointed out, we're focusing on skeletal muscle. And so the disorders that we're going to talk about are on that spectrum. The MCAT is not going to test you nitty gritty, which nerve innervates, this organ, like this, the heart or the, what
the different myotomes.
Exactly. Yeah.
That's a term you're going to learn about in med school, and you're going to hate it. Or at least I did. Yeah. So there's definitely something there. And I do just want to like do one last little mention that like acetylcholine is also a big part of the parasympathetic nervous system. And your parasympathetic is the rest and digest stuff. So you actually do release acetylcholine onto the heart to actually make it contract less because of the parasympathetic. So like the heart and smooth muscle, a little bit different than what we're talking about now. And that's just like one very clear example that acetylcholine makes your skeletal muscle, like your bicep, flex, but it makes your heart actually flex less often. And so that's something you want to be just kind of careful with. But like the whole reason we're bringing up this understanding this thing is because there are some very interesting diseases and disorders that could very easily show up on the MCAT. Probably the most common neuromuscular junction disorder is myasthenia gravis, which is a very interesting disorder where basically it's an autoimmune disease where you produce antibodies that bind your acetylcholine receptors on your muscles. So like I'm producing antibodies that will attack my own muscle receptors. Like, so if I release acetylcholine, it can't get into the receptor because there is an antibody in the way. And so this is a kind of an interesting thing where it is an autoimmune disorder, but it starts to have some really big effects on like how you are like flexing your muscles. It makes it harder. So typically with you have less active acetylcholine receptors. And so you have like less muscle activity. And most of the time, it's not like a complete paralysis, but like a weakening. And so like I can't get my muscles to fully contract because a lot of those receptors are blocked. And this is like you have this just kind of like weakness overall, even though you still do have the muscles. Now, what makes this even worse is over time, like your immune system is attacking those muscles, and you have like all sorts of this immune response, including like the complement system and a lot of other things. But you can actually end up losing muscle cells due to that autoimmune thing. So not only do these people have a harder time getting their muscles to contract, but all of a sudden they start to destroy some of the muscle cells they do have. And so you have less muscles and they don't react as strongly, they don't flex as strongly. And so this can cause some really severe weaknesses. And so a lot of times people with myasthenia gravis have trouble walking and just kind of like lifting heavy things. They get tired very quickly. And so there's all sorts of interesting tests. I know, Azaii, that you've been kind of like, in the realm of like med school. And so there's a lot of focus on those tests, but why don't you kind of illuminate a couple of those for us?
Yeah, I think it's really great that you highlighted how myasthenia gravis actually works, because we're not giving you guys these illnesses just so that you have something new to memorize, right? The MCAT isn't going to test you on, what medication is used to treat myasthenia gravis, or, how is it diagnosed, or anything like that. That's for med school. That's for later in life. Mm-hmm. But what we want you guys to be able to do is take what we're explaining and help it and have you use it to help you understand how things work. Right? Not just how the disorder works, but also how normal physiology, the body normally works. And then once you know what goes wrong, it helps reinforce what normally goes right.
Yeah. Well, the MCAT isn't going to ask you to understand how to diagnose or like the symptoms or the treatments of this. They will expect you to know what happens if an acetylcholine receptor is blocked. And so like this is like a great way to kind of understand that pretty in depth, kind of overall.
Yeah. So just to recap, right? So you have these antibodies that your own body is producing against the acetylcholine receptor. So if you think about what's happening, right, you have less of these receptors available. So acetylcholine, right, it's going to be present, but you're going to have a harder time getting that full contraction. So if you think about what might be a potential treatment, right, something that allows there to be more acetylcholine, perhaps, right? Because now you can have a more robust contraction. And if you leave that acetylcholine in there, right, and you don't do what Phil was talking about before and chew it up, then you're going to be able to maintain that muscle contraction. So one of the treatments is an acetylcholinesterase inhibitor, right? So hopefully that makes sense, because if part of the issue is you're not able to produce a robust enough contraction, you don't have enough receptors, right? You have some acetylcholine hanging out, but then if you now have a lot more acetylcholine in there, you can have that more robust contraction.
You'll be able to activate the receptors that are open a whole lot better.
Exactly.
And I think that's a really interesting thing. There's also some like symptoms of like people just tend to get tired, and like the longer the contraction goes on, the harder it is for them to keep it up. And so you can do like tests, like have them just look up for 2 minutes. And if they like struggle with that, and like as time goes by, then that like could be a myasthenia gravis indicator overall. But I do think like that, like blocking of acetylcholinesterase, that just means that you have more acetylcholine out there. What's interesting about that is, yes, that will strengthen the contraction, but it also is going to slow down the rate of relaxation, because that's how we relax the muscle, is like get rid of the acetylcholine and chew it up. And so if we leave the acetylcholine out longer, then it's going to be harder to contract. And so this is one of the reasons that there's some kind of like people who are on these medications, they are stronger, like before they were having this weakness, but it's harder for them to relax. And so like it becomes very difficult to like play the violin or piano, because like it's harder to get your fingers and muscles to relax as fast kind of overall, which is kind of an interesting thing. And that can also create some difficulties like with walking. And like, let's say before your muscles weren't strong enough, you weren't able to get enough contraction to walk. Now maybe you can, but like you take a step and it takes a second for like your muscles to relax a little bit. And so you end up with like kind of a different gait where you have to take slower steps, because it takes a little bit longer for your muscles to relax, and it requires some more fine-tuned control. But it's just kind of an interesting thing kind of overall, looking at myasthenia gravis and the treatment for it, and like understanding that acetylcholinesterase, that's also something the MCAT will totally ask you about. Like what is going on with the role of acetylcholinesterase and the acetylcholine like receptor and junction, which is once again why we're diving into it at this level.
And also something that's really, I think, interesting to keep in mind is that what we just described is a treatment for the symptom, right? And you can't reverse the damage, right? By giving an acetylcholinesterase inhibitor. There are some cases where some people, they actually have a thymoma. And so we know what is, contributing to this. And if you remove it, it can actually be curative, but that's not always the case.
Well, what's a thymoma?
Yeah, so that's just, so in your thymus, you have some maturation of T cells. And so basically just like a tumor, so to speak.
Yeah. So like thymus is like a big part of the immune system. And anytime you have a tumor, it's called like, we throw oma at the end, like a sarcoma, for example. So a thymoma would just be a thymus tumor that like, maybe that's why your immune system's going crazy, because it's hyperactive. So removing that might actually cure the myasthenia gravis.
Yeah, if you can find it. It's like, that's why actually, just fun fact, when someone gets diagnosed, they have to get imaging of their neck to look for one. It's an automatic thing, because you have a potential cure. So pathophysiology, why things go wrong and why they normally are right, is so important to figuring out not just like what you're learning, but also the treatments. So we strongly encourage you guys to dive into the normal, like what normal looks like when you're studying for the MCAT, as really great preparation for the rest of your life.
Yeah. And I think that's important also to just keep in mind. I'm like, a lot of students are like, wait, why do I need to know that it's a ligand gated sodium channel, right? Like, why is that important? Yeah. Well, because that means Like there's got to be something that binds. And so if you block that, that becomes a problem. And so I know it doesn't seem like it sometimes, but a lot of the basics of this like idea and like the content actually explains what's going on with these, kind of diseases kind of overall. The other thing that we want to talk about, it's like one of my favorite, is that okay to say one of my favorite toxins? I probably shouldn't say that. It's one of the most interesting toxins, which is botulism. And so there's botulism toxin, which is created by Clostridium botulinum, which is a gram-positive bacteria. Note that we're probably not going to go super in depth into it, but you need to know the difference between like gram-positive and gram-negative for the MCAT. We're not going to hit that here. We probably will hit that in a future podcast episode. But this toxin, and like, once again, what's really interesting about this is it's not the bacteria itself that is causing the problem. It's the toxin produced by the bacteria. And so it's the botulism toxin. And so what this does is it affects actually the neuron. So this is affecting things a little bit earlier in this pathway, right? Because myasthenia gravis blocks the receptor from the acetylcholine being able to bind it. Botulism toxin makes it so that the neuron can't release the acetylcholine. And so it's a very similar effect in that, like, all of a sudden you have muscles that are not working because we've kind of like slowed or stopped what's going on with them. But the botulism is actually a little bit more of a severe thing because you're not releasing any acetylcholine. And so it's not that your muscles are weaker, it's that they are completely like uncontractable, like they're paralyzed. Those muscles will not fire because even if you send a signal down that neuron, it will not release the acetylcholine. And so this is actually something that used to be a really big problem back in the day, back in like, pilgrim times, because botulism, like Clostridium botulinum as a bacteria, is actually anaerobic. And so most of the time where this would grow would be in canned foods that don't have access to oxygen. And so the bacteria would kind of like grow and divide and do stuff in there. And what's interesting is like the bacteria would generally also just die because it could not survive in that environment for like an infinite amount of time. And so even if you can something and it had some of this bacteria and it grew, and then even if that bacteria died, right? And so there's no bacteria in there, the toxin is still around. And like the stuff that this bacteria produced. And so even though this jar of like green beans might not actually have any of this bacteria in it, it might still be dangerous because of this toxin. And this is one of the reasons that it's really important. If any of you guys are canning food, while you do this, like you boil it and you heat it up and then you put the lid on it, because boiling the stuff inside will kill the botulism or the botulinum bacteria. And so you like heat it up to the point where the bacteria can't live, and then you close it up and seal it. And then you cool it down. Like if you heat it up and then you cool it down and then you put the lid on, like some bacteria from the air might have gotten in there, and like that can all of a sudden be a problem. And so the real issue with this, everyone's like, okay, well, all right, so it's in this food and like it makes it so like my legs don't work. I mean, like the toxin we're going to deal with eventually. Well, the real problem is your diaphragm, which controls your breathing. And so that if you paralyze that, you will stop breathing. And so botulism actually killed tons and tons of people back in the day. We don't really worry about that today because not many people are canning their own food. Like the number one cause of it was improper canning. And that doesn't happen that often now. But it is something that's super interesting. There's also this like toxin, it has this ability to completely like paralyze things. And that seems like that's what makes it dangerous. That's what makes it fatal and like makes it able to kill people. But like we could actually hijack that and use it for different ways. Like if we need to paralyze a muscle, this can be a really good way to do that. Like, so this botulism toxin is commonly used as a drug or as a treatment for things as well. Just to like, kind of scrunch that word together, botulism toxin. And some of you guys may know this, like what we call that is Botox. And so I'm sure everyone has probably heard of Botox, which is that thing you like inject into your, like, you know, your brow and like your crow's feet and like the edge of your eyes. What that does is it just paralyzes those muscles, so you can't contract them and end up with wrinkles in your like forehead. It does make it so you can't contract them. And so I remember when Botox first came out, man, I feel old. Like as a treatment for like making your skin prettier. That would like, there's all sorts of like movies and TVs that were kind of making fun of it, where someone would get it and like all of a sudden like they can't like express any emotion at all, and like they can't smile or like move their eyebrows. And there is something to that. If you inject enough of it into the wrong places, all of a sudden you lose all expressivity, which is kind of funny in those comedy movies. But it's also used for a lot of other things. It's not just like beauty things as well, because that like releasing of the neurotransmitter is also related to other things like sweating. And so if somebody has excessive sweating, this can help with that. If somebody's having muscle spasms, paralyzing that muscle can totally help. It's also had some really good uses with migraines as well. And so just kind of an interesting thing, this thing that used to kill like thousands of people is now used to make ourselves look prettier and make me sweat a little bit less often, or help with migraines or muscle spasms or things like that. And so we're hijacking this thing that used to kill people and using it to help people instead, which happens a lot in medicine. But like, I think it's always something that I always get excited by, like looking at like the way that we can hijack some of the machinery with HIV or different things like that. I don't want to get into the gene editing stuff because I have a whole lot of, it's super interesting, but it happens all over in medicine. And like, this is why the better you understand it, the more you can use it overall.
Yeah. And really quickly, this is something we don't worry about as much in adults, but we still do in infants. So things are a little odd in infants because they don't have the same immune system as we do. They're not as, well prepared for whatever onslaught they might, get of microbes and toxins and whatnot. So this is actually, botulism is the reason why we don't give honey to infants, because the spores can actually be found in honey. But infants don't have the microflora, right? The gut microbiome that we do. And so in that particular case, they can actually, the bacteria can colonize and release that toxin. So we don't worry about it in adults. We still try and avoid giving our children honey. So if any of you are new parents and you haven't been told that just yet, don't give your children, don't give your infants honey or your newborns honey. Okay.
That's super interesting. And I know a lot of people like, wait, this thing is found in honey, I can't eat it. But like, remember that the bacteria is not the problem. It's the toxin that's the problem. And so if the toxin like reproduces enough and produces enough of the toxin, that's when it gets dangerous. And in newborns, like that bacteria can grow in the intestines because their immune system is just weaker. And so like the toxin will be created all at once, like inside the newborn. The toxin isn't in the honey, it's made while inside the newborn, which is once again kind of super interesting to like understand kind of how this all fits together.
Yeah. And I think it's really cool that we are also taking the time to think about, okay, where in signaling does this occur? So first we had myasthenia that was at the level of, the receptor on the muscle. We have botulism and the Botox occurring at the level before the acetylcholine gets released. There's actually another one that also occurs at the level where it prevents the acetylcholine from getting released, but it seems to have the opposite effect. And that's tetanus. So with tetanus, so in this case, it's the Clostridium tetani bacteria and the spores. And so if you think about, when does someone say, oh, did you get your tetanus shot? So if you stepped on a rusty nail, right? So that nail has created a deep wound in your foot. And so these anaerobic bacteria, they like it. They go, right? And they're actually going to bind some of the receptors on your peripheral nervous system, but then they're going to work their way back up to your central nervous system. And so they're going to do something really similar to what botulism toxin, does. And that's, they're going to prevent the release of a neurotransmitter at this junction. And so in this case, it's actually going to be in GABA and glycine or inhibitory neurons. And so if you think about before, acetylcholine was going to promote muscle contraction. Now this toxin is preventing the release of a neurotransmitter that's going to inhibit it. So what would you expect to happen if you prevent inhibition, right? Contraction. You're gonna have a whole lot of contraction. So on the opposite extreme of the paralysis, here you're just going to have a lot of muscle rigidity. So your muscles are just going to be very tense. You're going to have spasms, right? So that constant back and forth. You'll actually, it's kind of very intense. If you ever look it up, the people will have like their entire backs fully arched. And this, there's a specific smile, there's like locked jaw. So there's just a lot of tension. And it's odd because I know I always struggle, like, wait, why is their back arched if it's muscle contract? Like, why wouldn't you go down? If you think about your back, right? It's like pinching and then it leans back. I just felt like it was very important to
like you will
that.
flex the muscles that like bend yourself forward, but your back muscles are stronger. And so you're flexing both at the same time. And like the ones in the back are more powerful. And yeah, like you also said smile, which makes it sound like, oh, this sounds great. But no, it's because all the muscles in your face are like contracted like insanely, and like it pulls everything back. And it is super intense. And it's something that, like, at first when I like looked into it, I'm like, oh, like that sounds interesting. I wonder if this would be a good way to like, like make my muscles stronger, right? It's like, oh, it like increases contraction. So it'd be like exercising. But no, this is like, imagine having a cramp in your leg. That's like a muscle that's like contracting when it shouldn't be. Now imagine that cramp all over your body for hours, right? In your face and your neck and your back and your arms and your legs. And like, it's incredibly painful and difficult for people who have it. Would not recommend 0 out of 10 having tetanus toxin. But like, I noticed you said it was Clostridium tetani, which is the bacteria. I think it's also important to like recognize that the botulism was Clostridium botulinum. And so these are both caused by like the same family of bacteria, but they have opposite effects. One causes like complete paralysis and one causes complete contraction. And it's like they're two sides of the same coin. And so it's super interesting kind of overall. Yeah,
it really has so much to do with not just like what is causing the, like which bacteria, but also like, what are they affecting? Like which neuron type, right? Which signaling pathway are they affecting? And so I love that you bring that up because it's so important. I know that personally, I have often found it difficult to sit down and be like, why do I care about this particular pathway? Like, what difference does it make? I need to memorize like 5 or 10. What does this one matter? And having something that you can tie and say, oh, okay, this is why this one, matters. This is why it can be important. This is where it might come up. Can be really helpful. And note that both botulism and tetanus, they are fatal if they are very, fatal if untreated. So those are one of those do not miss diagnoses. You do not want to miss it if someone comes in with it. But the reality is we have treatments. So in the case of botulism, we didn't talk about this, but we have like antitoxins. With tetanus, we have, antibiotics, antibodies. So just little things that are interesting to remember. And we want you guys to, not necessarily because you'll be directly tested on them, but you'll be tested on something related to them on test day. And they're just really helpful to start learning how things work and cement how things work.
Yeah. Not only that, but the MCAT could totally throw a passage at you about any of these topics and about how these things work. I think it's kind of interesting. The botulism antitoxin, basically that is an antibody that binds your antibodies. And so it stops the antibodies that bind the acetylcholine receptor from binding them. Or not the acetylcholine receptor, the presynaptic neuron. So it's an antibody that binds and hides your antibodies. And kind of like a weird competitive inhibition sort of thing. And so that's something that could totally be a passage on the MCAT. I also think it's interesting, like the bacteria that creates the tetanus toxin that bacteria just likes to grow in rusty metal. Like there's something about oxidized metal. It likes to grow in that area. And that's why, like, if you ever step on like a rusty nail, all of a sudden, like your primary care provider is like, ooh, when's the last time you got a tetanus booster? Because we can give you a vaccine to stop this bacteria from being able to grow like in that wound in your foot and stop it from producing this toxin. We don't have a vaccine for botulism, but there is a vaccine for tetanus. And so I think it's also interesting kind of like seeing that the MCAT could totally test about vaccination. That's a topic that they love to test about. And so understanding like tetanus and kind of its role in that is also something that's highly MCAT testable.